The overall goal of this procedure is to visualize the vasculature of Xenopus tropicalis tadpoles.
This method can help answer key questions in the field of cardiovascular biology, such as how angiogenesis is regulated. The main advantage of this technique is that blood vessels in an intact wild-type embryo can be visualized by a simple fluorescent dye injection.
Begin by using a microloader to fill a tapered glass micropipette with approximately 5 microliters of clear diacetylated LDL solution, taking care that no precipitated particles are loaded. Under a dissecting microscope, use a fine pair of number 55 forceps to clip the tip of the pipette to adjust the volume of the uploaded complex, and set the pressure-controlled injection system to eject approximately 5 nanoliters of solution per injection. Then submerge the pipette tip in 0.04% MS222 in 0.1x modified Barth saline or MBS in a custom-made Silgard mold. Next, transfer a Xenopus tropicalis embryo to the Silgard mold and confirm complete sedation with a lack of response to dorsal fin stimulation. Under the dissecting microscope, load the embryo ventral side up into the V-shaped indentation of the mold in a slightly oblique position. Tightly fit 2 0.1 mm Minution pins to a pin holder and use one pin to puncture the skin over the heart, which should be visibly pulsing.